I think it not success for industry now, it's just in labor. Also, besides the chemical methods, there are also some biological methods to get the chitin from the collagen shell. We can use the enzyme to remove the protein, and also can use organic acids and producing microbead to remove the calcium. We do this in the label. It also has high activity. They can success to remove the protein and calcium, but also the cost is much higher than this traditional chemical methods. So, but we set up a small scale production line, not a big large scale production line, but a small scale line is okay. And but I think in the future, maybe this biological system will be the play methods to extract chitin, because it's more clean and it's not more pollution. The second point about the polysaccharide from the seaweed, from the alga. Take this arginate, for example. This is the process of the extraction of arginate by chemical method from seaweed. The raw materials usually we treatment, and they use acid, use this base extraction, and we to use this to optimally to change the different index to get a more index to optimization the process, and still need to use more alcohol to get the final products. This part is not like the chitin part. Not like to extraction the chitin from flavor. Is not so many pollution, but also need the base and also need the alcohol, and more important, this state needs lots of water. So it also will make pollution. And so some green approach will build up, like to use the organic acids, not so strong, and to remove the water or use the water recycle to do this. But in my mind, now also this traditional chemical method is the main method in the industry, in the industry production for these green methods. Still to education. The third part is about the microbead polysaccharide. Take the fungi cell wall polysaccharide as the example. The fungi cell wall is quite complex, and it's also very important for the fungi to survive. It can maintain some normal cell metabolism, interchange, and osmotic pressure, and also when it can have important function when the fungi to attack the plant or attack the animals. The fungi cell wall also is very important. This fungi cell wall can be divided into two parts. One part is the out part can be water soluble. The inside part can be water soluble. These are different fungi cell wall from different fungi. From this cartoon, you can see there usually there are two parts. But most of this picture are not the true picture. It's just based on the information we know. I think now we still even for this saccharomyces crude, which was well studied for their cell wall, the true cell wall, how to build up is still unknown. It's just this kind to show based on our knowledge. But you can see it's quite complete. To instruction and identify the fungal cell wall, usually there are three steps. First is the sample collection and the crude polysaccharide extraction. Usually there are several methods, like the water extraction, dilute acid or alkali extraction, and the microbial assisted, enzyme assisted, and also other extraction methods. Sometimes it can be merged, sometimes it can use one by one. This is our first step. The second step is the purification. Usually use HPLC to what they separate different polysaccharides. And the third step is to, if we want the purified polysaccharide component, then do the structure characterization. Usually there are some different analysis methods, like the spectroscopy, the HPLC, the methylation tissue mass, the NMR, and based on all this information, it can conclude and make the structure, like this, to make a conclusion. But it's just based on the information. Still, I think on this part, there's still lots of work need to do. The last part about the polysaccharide is from the plant cell wall. Yeah, it's also very different and complex of the plant cell wall. But there are three major plant cell wall parts, as we all know: the cellulose, the hemicellulose, and the pectin. The plant cell wall is also very important for this growth, development, and this resistance to the fungi. And also the small part, the hydrolysis products of this plant cell wall can be elicited to promote plant immunity, which I will talk on the day after tomorrow, the last lecture. The cellulose, usually they are the same. The cellulose is a very simple polysaccharide built up with glucose. But most different plants, the hemicellulose and the pectin are different. These are some message from published paper, but I can't say all of them are right. Maybe in future some new paper where I found something strange, not like this. And now this is commonly seen in the decocandidates. The xyloglucan is a main hemicellulose component. The monocose, the xylem is the main hemicellulose component. These are the different polysaccharide structure. Yeah. The extraction is the same like the fungi cell wall, but this picture shows different. Some are very advanced methods to study the cell wall polysaccharide, especially this one. This is NMR. This solid NMR is a very advanced machine technology, which I think this method can give a true information of the complex link polysaccharide. But unfortunately, these are not popular equipment and very expensive. Also, in my institute, we don't have this facility. I think in USA, some university have this. A famous professor called Wang Po, also in Chinese American science, he published also based on this equipment. Just talk about role. Just talk about the polysaccharide structure is very complex, but in the divide, it also are simple carbohydrates. This part is glycoconjugate. These are complex carbohydrates because it not only have the carbohydrates, it also have other compounds like glycoprotein, proteoglycan, and glycosinopholipid. And also for the glycoprotein, it can contain the N-glycan or O-glycan, but all these different glycoconjugates are the glycan will aid by glycosyl transferase to these different protein or lipids. Yeah, I will give a quick introduction of all these compounds. Yeah, I think this picture, you can see this picture in many books, is about what is about the complex carbohydrates. Yeah, this is about the N-glycan. Why it is called N-glycan? Because these are linked to this amino acid with this N in the protein. Yeah, this the N-glycan finally here. The N-glycan's biosynthesis pathway is also complex pathways. By more than 20 enzymes step by step from the protein to add different monosaccharide step by step. Finally, it can be like this glycoform. But this glycoform is different in different biology. In this picture, in different organisms. Yeah, and this also the N-glycan change to from in the factory there are no N-glycan, and in the yeast though the glycan is longer, but they only contain 2 monosaccharides. In plant, and then in animal, finally in human, the N-glycan will evolution complex, and also have the organism's spatial character. For example, in plant, there are xylose, which are evident in plants. In human, there are these salicylic acid. For this part, I just give a quick review because we focus on polysaccharide on all the lecture. Besides this N-glycan, there is also another glycosylation called O-glycan, which link to the S or T amino acid in the protein. It also have different kinds of O-glycan. Some is a long one. Read this. Some long one is also the most common in the organic glycosation, and also some short one. Most famous is the O-glucanate. I just show the O-glucanate silation, show this O-glucosilation. I think most of you know this phosphorylation, which is very important on protein's function. And this O-gluconesis base can be harmony with phosphorylation on the same point. On the same point in the plant. Even if the O-gluconation on this amino acid, the phosphorylation will count. They can't appear to this side. So these are very important glycosylation, which nearly half of the protein will have this kind of glycosylation. And more interesting, I think you all know that enzyme is quite selective reaction. But for this O-glucosilation, for the OGT, which is transfer this gluconate to the protein, and for the OGA, which is cut this gluconate from protein. But in every organism, for example, in our human, we only have one OGT and one OGA, but this OGT can transfer this gluconate to nearly all the protein from the naked protein to be the glycoprotein. And this OGA can cut this gluconate from all the O-glucosylation protein to be naked protein. Take charge of this very important function, and this can be glucosylation on nearly 10,000 protein, but only controlled by 2 enzymes. So these 2 enzymes are very important and lots of research. The Professor Gerard Hart, which first identified this O-gluconate and OGT, was the most important, most possible person to win the Nobel Prize on this glycobiology research aspect. Yeah. Then about the proteoglycan. These are some proteoglycan. Both the glycoprotein and the proteoglycan, they are built up with glycan and protein. Why they are called glycan proteins and these are called proteoglycans? Because the glycoprotein, the most is protein, the glycan is least. But for the proteoglycans, most are glycan, the protein is least. So the name is different. Yeah, I don't read the definition, but just this glycosamic glycan, the GAG, is a very important proteoglycan, which in nature and which is also an important medicine. There are different, there are typical GAG structure for this. Hyaluronic acid, I think the goals are very known. This is also for skin, for good for skin, and sometimes it can be injection for a beautiful change. This GAG also like N-glycan is have a very long biosynthesis pathway. Also different enzymes will have very strict function to add different monosaccharides space by space about the this part is quite complex. So in my label, we never do research on glycolipids because the glycol is very complex. The lipid also very complex. So I do research this part, but this is also very important. For example, the GPI alcohol protein is very important for nearly all the signal transduction. Yeah, these are also the definition and also the biological function. Most important about the cell structure and function, and yeah, they will show one example in the blood group antigens. Yeah, our blood group are ABO and AB. Their antigens is the difference on the antigen of cells in the last monosaccharide. The group A is the GANEC. The group B is the galactose. This place in the two monosaccharides is different. And this different is controlled by the different UGT, the different glycotransferase. The group A have this A enzyme. The group B have this B enzyme. The group AB have both enzymes. The group O don't have both enzymes. So this is important to change the group is a small change from the monosaccharide. This is an example of the glycolipid important. Also, the glycobiology information can be learned from this good book, The Essentials of Glycobiology, the first edition, 1999, the second, the third. Now is the fourth edition in 2022. It can be downloaded all the books in the CPI. So if you have some interesting on the glycobiology or carbohydrates, I suggest you can read this book. Yeah. Later also I show some examples of good glycoside articles, or from CNAs, Cell Nature and Science. Yeah. This is one aspect is about the polysaccharides are divided by count microbe. These are manual. These are pectin. Yeah, these are pectin. In maybe 10 years or 8 years before, they published lots of this paper. They got microbe to how to use the different polysaccharides. This is one aspect. The second aspect is that I just talk the glycosylation. For example, this part is about polysaccharide biosynthesis, and also this part is about the glycosylation important enzymes, the structural research. Both the polysaccharide biosynthesis and the glycosylation pathway in the human, in all the organisms is very complex, and most of the enzymes don't study well. So these are structural research on these enzymes related to polysaccharide and glycosylation pathway. This also published a lot of paper. Yeah. This is also an interesting paper. Yeah. At first, we just think this glycosylation to glycan can only on protein. But this paper by Bertozzi, the Nobel Prize winner, female scientist, she found that on RNA they also have this glycosylation. So both the glycosylation from protein lipid to RNA is quite interesting fun. So it's also published in Cell before she got the Nobel Prize. I think the paper also helped she to got the Nobel Prize. Also, this part also seems to research which enzyme to add the glycan to RNA, which enzyme to catch the glycan from the RNA is still unknown. I think who identified this also can publish CNA.